Welcome everyone, and welcome the world, whoever's watching live stream. So I'm going to give 2 lectures, one this week and then one next week. And I'm pretty sure they're swapping MPG to be 8:30 to 10, and then the primer will be 10 to 11 because of the schedule, the constraints of the MPG speakers. So if you want to come to part 2, it's actually 10 to 11 next week, I'm pretty sure. So the 2 parts are really talking about, with a moan in the background, gene disease validity is what we're going to talk about today. And then next week, we're going to talk about variant pathogenicity. But today's lecture is going to be basically the first step is before you can assess the pathogenicity of a variant, you have to know if there's enough evidence that the gene is actually implicated in disease. So that's what we're going to talk about today. There's some nice reviews that are out there looking at Mendelian genes. And I should premise that we are largely going to talk about Mendelian disease gene implication, less so around complex traits and pharmacogenomic associations and other non-Mendelian phenotypes. And this is a review from Mike Bamshad's group where they looked at all of the genes in the genome, and they were able to assess that about 15% have been implicated in Mendelian disorders. About 3% are Mendelian conditions mapped to locations in the genome, but for which the genetic etiology has not been identified. About 30% predicted to cause embryonic lethality and therefore not ever show up as a phenotype in humans until we can do better prenatal and early development analyses. And then about 52% of genes for which we really have no idea whether they would cause a Mendelian phenotype or might be not disease-causing if deleted. A more recent look at Mendelian disease implication was a paper led by Kim Boycott through ERDIRC, the Rare Disease Consortium across the world, and really looking at the gene discoveries over time and also separating by approach. So the red is traditional approaches using linkage mapping and then candidate gene analysis in the locus, whereas really in the last few years, almost all of the analyses have converted over to whole exome and genome next-gen sequencing approaches. And there is some question as we see the decline. We're hitting that stage where it's the hard things that are left, where it's very difficult because it's not it may not be a clear, nice loss of function mutation sitting in the middle of the gene, and it might involve more regulatory elements and deeper intronic variants and things that are harder to deduce their impact. And that's probably why we're starting to trail off as we get into this harder stage of discovery. I modified a figure that Daniel has shown in the past, really thinking about the comparison to gene-level evidence versus variant-level evidence. So we have mutations that are absolutely pathogenic in genes that are well-established cause of disease, like the Delta 508, most common cystic fibrosis mutation in the CFTR gene that we know is the CF or cystic fibrosis gene. So both high-level variant evidence, high-level gene-level evidence. We also find plenty of variants of uncertain significance in genes that we know cause disease, like a VUS in the CFTR gene. This happens in every gene that we know are Mendelian genes. We also have variants we don't understand in genes we don't understand, the VUS in the goose, as some people say. This category, however, cannot exist. You cannot say that there's a variant that is pathogenic if the gene has not been implicated in disease, because the term pathogenic means causative of disease. You could say I found a deletion in this gene that clearly wipes out the gene, but you can't say it's pathogenic because that implies a disease causality. So this category really can't exist other than just saying I've wiped out this gene because there's a deletion or something. When we do clinical interpretation of the genome, and we've been doing this in lots of particularly healthy patients in the MedSeq study or healthy newborns in the BabySeq study, and we go sequencing, a genome in this case 5 million variants, or an exome, 50,000 or so, and then we filter them against databases where people have reported pathogenic variants in either the HGMD database or ClinVar, or it's a novel loss of function, predicted loss of function in a gene implicated in a Mendelian disease, of which there are probably about 5,000 out there published as Mendelian disease genes. And we throw on a basic frequency filter to say, just get rid of the really high frequency junk. We find about 100 variants said to be pathogenic in Mendelian disease genes. But then we go through a process of excluding things, looking at the gene level evidence, realizing there isn't good evidence, and throwing all those variants out. Some variants are fairly frequent, yet been claimed as pathogenic before the age of Exec and Nomad, in essence. We can quickly throw out those and stop looking at them. And then we have a whole bucket that are just extremely rare, but still insufficient evidence for disease causality at the variant level. And that gets us down to only a handful of those variants that actually can be said to have sufficient evidence for both gene-level disease implication and pathogenicity at the variant level. So that's a lot of work to go through, and a lot of these are gene exclusions, where you don't even get to the variant level, you just look at the gene and all the evidence and go, it's not enough. For example, here's a baby-seq exome case that we had last year, healthy newborn, born in the Brigham well baby nursery, no family history disclosed, and we discover a predicted loss of function variant, a frame shift, in the VCL gene. So we're going to ask questions. First question, is VCL a disease gene? And then the second question, once you say yes to that, is it the variant actually pathogenic, the individual variant? Of course, in baby-seq, we also have the question of, is it actionable? Should we return this to a newborn who's ostensibly healthy? So we're going to focus on this question. And in this case, this gene was reported as a dilated cardiomyopathy gene with loss of function as the mechanism. So at first glance, you're like, whoo, maybe this baby's at risk for dilated cardiomyopathy. But then you go through the evidence, and there was a zebrafish model using transient knockdown with morpholinos that just showed a DCM-like phenotype. There was a knockout mouse with early sudden death. It didn't actually have signs of cardiomyopathy, but had rhythm disturbances. But the mice who survived longer did develop a DCM-like phenotype. So there was some, animal model data. The first gene or variant implicated in humans, a in-frame single amino acid deletion in a patient with DCM, later was refuted once that variant was found to be in 0.5% of African chromosomes in Exec. So the first human disease claim was refuted. There had been a few more papers that came along finding missense variants in this gene with no segregation data. So you look at that and you really go, there is insufficient evidence that this gene actually causes a human phenotype. And so we would not report that. Now the question is, how can we do, I sort of, made my argument and showed you the data, but is there a way we could slightly more systematically do this? Through a quantitative or even semi-quantitative approach, because we want to distribute these approaches to everyone in the world to look critically at gene-level evidence. So through the ClinGen program, which is developing resources to define both gene-level evidence and variant-level evidence and actionability, and providing that freely to the community, we started to address a systematic approach to gene-level validity. And these genes, well, I'll go into that here. So the approach here is defining the criteria to assess gene disease validity. It is a semi-quantitative approach, and you'll see what I mean by semi-quantitative. The challenge here is that you can't really just take a statistical approach, because you're dealing with a whole bunch of different pieces of evidence that are difficult to combine in a multi-likelihood model, and a lot of which is really not quantitative and very judgmental. So we had to take some degree of a non-quantitative or semi-quantitative approach to this process to deal with a multitude of different types of data, much of which is not quantitative. It allows us to methodically classify validity of a gene-disease pair. And now keep in mind, genes are often implicated in multiple diseases, and so you have to assess each one VCL for dilated cardiomyopathy. Maybe there's another claim that comes out that says it's a hypertrophic cardiomyopathy gene. Separately evaluate that evidence, and so on and so forth. It is not intended to replace well-established statistical thresholds used for GWAS or other validated methods for multifactorial disease risk. It is mostly targeting Mendelian diseases where we have insufficient quantitative approaches. Feel free to ask me questions, by the way, along the way. So this is the overarching framework, and we also came up with terms, kind of like in variant assessment, we have pathogenic, likely pathogenic, uncertain significance, likely benign, benign. We came up with terms to describe the level of evidence that we're talking about in a terminology manner. And so we have definitive, strong, moderate, limited, no evidence reported in humans. Maybe there's a mouse model, but no human evidence. Or there's conflicting evidence that may dispute the claim or entirely refute the claim. So these are the categories that we use. There's a high-level sort of description of that, but I'm going to actually go in a little more depth into what we consider these categories to mean. I will give you an example of the challenge here and thinking about how we might use this information in the clinical arena. So this could be used to guide inclusion in disease-targeted gene panels. For instance, we might only use genes with moderate and strong and definitive in a diagnostic panel, and exclude the limited evidence ones for really discovery purposes or when you're doing exome and genome, things like that. Only look at definitive and strong when you're doing predictive tests in a healthy individual. You really want to make sure you know what that gene means. And just by way of the challenges here, this is actually a figure that shows the number of genes included in clinical panels for cardiomyopathy testing across 14 US clinical labs. So you've got, some labs with 90 genes, other labs with, this is 15. So huge variability. And that's in part because people aren't systematically looking at gene level evidence and making good decisions about what gene has enough evidence to be put into a clinical test. So that's what we're trying to help with in some degree. So this is kind of a little bit of the framework. So we look at genetic evidence and we score it according to point score. We look at experimental evidence. We sum up those two. And then we also look at replication and period over time to allow the community to develop data that may dispute a claim. And then the point values that we're adding up are then going to land you in limited if there's 1 to 6 points, moderate if there's 7 to 11, strong if there's 12 to 18. We get to definitive if the data has been replicated, repeated publications, and has been at least 3 years to allow the community to mount conflicting information if it chooses. So that's kind of how we go about doing this. There's two smaller figures, and we're going to go into these in depth so you can actually read them, that look at genetic level evidence and functional evidence. And, you can now see what I mean by semi-quantitative. We're, using scoring and points to sum up evidence, but there is an arbitrariness to this. Like, how much do you think one missense variant that polyphen says is damaging should count for? So a lot of this in some ways was iteratively developing the scoring, seeing how it came out, using experts who really understood those diseases and comparing and then modifying over time. And so that's sort of how we came up with the point scoring. Is that there is a fair bit of subjectivity and expert opinion in how to think about the points. And every time you go to assign points, there's also subjectivity to this. So at that level, we have a stage where experts review the data that's been aggregated and the scoring that's been applied and say, yes, I agree with that, or no, I don't. I didn't count this animal model because that phenotype doesn't really match, what I think is the human disease. I don't think that's good data. I would decrease that scoring. For example, that's how the experts might weigh in. So in our framework, we developed it, we validated it, or we applied it to 33 different genes. We had 2 different curators test out each of those 33. We went through expert review and we measured concordance. So concordance between the curators was 94%. Concordance between the curators and experts was 88%. 4 genes differed by only 1 level. We excluded 2 genes were classified as no evidence and were excluded from the % concordance. All of the classifications that we are doing are on the ClinGen website. On the front page, you can go to the search our knowledge base, type in your favorite gene, and if it's been curated, and this is the list of curated, now that includes dosage curation also, so on the website right now for gene disease validity, there's still only a couple hundred genes there yet, and we're working on getting more of what we've done uploaded. But if your gene is on there, in this case ACTA2, it will be shown for the different gene level curations. Perhaps actionability has been done, dosage sensitivity from our cytogenetic group, and then here is the gene disease validity, which is what we're talking about today. And you can see ACTA2 in association with aortic aneurysm scores as definitive as reviewed on September 16th, and you can click on that report and see it. Now, it's very critical, the date, right? Knowledge evolves, things change over time. And the good thing is things that hit definitive, we don't feel we need to continually review, but things that are lower than that, you have to go back because new papers will be published. So once you click on that view report, you'll see the way the scoring was done, the publications that were done, and the final and the expert reviewed classification, the date it was done. Over time, there'll be even more detail that will be provided, but this is sort of the summary level right now. One of the geneticists in my group for our BabySeq study applied this in more of a high throughput fashion without doing all this point scoring, and curated 1,514 genes that are a little more well established out of the 5,000. And this is what she came up with in that process. Third were definitive, a third strong, 16% moderate, 16% limited, and a few that were conflicting. She also curated age of onset and penetrance, because those are factors that determine whether we return results to a newborn. But this gives you a sense. Now, this actually looks much better than the reality, because she picked the best 1,500 genes based on some publications. It's actually not as good, and here's an example of going deeper into a particular disease area. So our hearing loss group curated 163 genes for gene disease validity. 44% were definitive, 7% strong, 27% moderate, 18% limited, 4% no evidence, conflicting, etc. So that gives you, and that same thing sort of looking at RASopathies. This is now being done across many disease areas, cardiomyopathy, aortopathies, cancers, neurology, epilepsy, etc. So a lot of different expert groups that we've convened that are now diving into gene curation, and that data is starting to get uploaded to the website. So if you have a gene that's limited, what happens to it? Like, does it just sit there and limit it in perpetuity? So, key question is, how do we build evidence over time? So here's an example of a case that presented at Brigham Hospital. Some of you may recognize Anthony when he was in his younger years as a resident. He was involved in this case along with Mike Murray, who's chief of genetics at the time. So this is a patient who had distal arthrogryposis type 5, a well-described Mendelian phenotype, but for which the gene was unknown at the time. And it has, a distinct and varied type of phenotype that involves the skeletal system, muscle, face, ears, eyes. It's actually reasonably straightforward to diagnose the phenotype. However, the gene was unknown. It had been described to show dominant inheritance as well as de novo occurrence. And this particular family that was being seen at the Brigham had one proband with healthy parents. So there was a suspicion, obviously, that this is probably a de novo occurrence. That's the perfect scenario when you're trying to do gene discovery, because you can look for de novo occurrence. And sure enough, about the rate that we would expect for de novo variants in the coding sequence, you typically see about 1 de novo variant coding sequence. And in this case, 2 de novo mutations were found, one in the ACSM4 gene, which was kind of quickly ruled out based on the fact that there are nonsense variants in the EXAC, or at the time it was ESV, with fairly high frequency. However, this piezo2 gene, the variant was, there was no, suspicion about this gene. It was, I think, constrained. Actually, we didn't have constraint scores back with ESP. But all that was known about this gene, it was a mechanosensitive ion channel. It's never been implicated in disease. So it's a great candidate because you found a de novo variant, but it's not sufficient for causality, right? If I sequence you and your parents, all of you in the audience, we'll find about one de novo variant in each of you, right? Doesn't mean that variant is causing some disease. So if we go sequence people's exomes with diseases and we find what we'd expect was about one de novo variant per exome, that doesn't mean it's causal. So how do we implicate this gene? And I should mention, and this will become clear in next week's lecture, when we see a de novo event, without known association to phenotype, you really can't conclude anything. It is uncertain. When we see it in a gene with an existing phenotype, yes, Yossi?
When you say, if I sequence all of you and your parents, I'll find one de novo, would that, I mean, if you do some sort of functional annotation on that de novo, would it look like a causal variant, or would it look like a, I mean, if in that thought experiment, would it look more like a kind of a silent,
right.
As opposed to, I don't know, I mean, of course you didn't tell us about that one, but is there an informatic observation that you could make on that?
So it's a great question, and in fact, when we had this scenario, and we had no other family, no other sequencing data, variant data, the first question was, well, let's turn to functional analysis, right? And so, in fact, there was one guy in the world who studied this gene. So we reached out to him and said, and this was led by Shamil, said, could you put this variant into your functional assessment, paradigm for this mechanosensitive ion channel that he'd been studying, and see if it causes an impact to the function of the protein? So that absolutely can be done, and you could demonstrate that it functionally impacts this mechanosensitive ion channel. But it is a separate leap to say that disruption of a mechanosensitive ion channel therefore causes distal arthrogryposis type 5. There are some phenotypes where the biological impact is a closer argument to the gene function. So for example, if you have a biochemical, inborn error of metabolism, where you understand that it's due to a defect in this enzyme, and then you go find a mutation in the enzyme and show that enzyme level, and you understand that biochemical pathway, you see a buildup of the substrate and a lack of the metabolized product, and you can make a very convincing argument for why your assay that shows disruption of this enzyme would actually lead to the phenotype of the patient. Those are more convincing stories. But in this example, when your function is a mechanosensitive ion channel, and you're trying to make the story of, well, how does that cause, spine stiffness and prominent ears and ophthalmoplegia? Like, it's a harder argument. For sure, disruption of an ion channel, which does lots of things, could cause this varied phenotype, but it's a leap, right? So I would argue that in this case, showing functional disruption of that gene would be insufficient to actually implicate that in disease. It's a piece of evidence, but perhaps not sufficient. So that's just an example. Just back to this story, when we see de novo occurrence, if the gene's already implicated in disease and you understand the mechanism, which is, then there must be an auto advance going on here. It's easier to call a variant pathogenic, but if the gene has not been implicated in disease and you just see a de novo occurrence, that is insufficient evidence to implicate that gene. Okay, so this was that guy, the one person in the world who studied this particular protein. What was critical here, however, was not getting him to show functional disruption. What was critical for this disease was a second family. And what happened was we called up this guy and said, could you put this variant into your system? He said, sure, happy to collaborate. And then a week later, a group in Canada called him up and said, hey, we see you're the only person in the world who studies this gene. Could you put our variant into your system because we found this de novo variant in this family with distal arthrogryposis type 5. Woo! That was the evidence that led us to bring that story together and led to the publication of piezo2 as the cause of distal arthrogryposis type 5. So it was really that second case where you could show an exact, extremely rare phenotype, both due to de novo mutations in the same gene. Without that second family, you're just sitting there, not enough evidence. He did end up showing functional disruption of the protein as well, but as I said, that would have been insufficient in my mind. So we've tried, it was just lucky that this group in Canada happened to call this guy and say, hey, I have a family too, and lucky that there was only one guy in the world that studied that protein that brought these groups together. But we, a number of years ago now, decided we needed to scale this in a more systematic approach, so we launched what we call the Matchmaker Exchange. And the idea here is you have a patient with phenotypic features that you code in using HPO terms, human phenotype ontology. These are standardized, structured phenotype terms. And then you put in your gene candidates. Maybe it's one gene candidate, or maybe it's two. You have two de novo variants, and they're both good candidates. So you put your gene candidates in. Somebody else on the other side of the world puts their patient in with their phenotypic features and their candidate genes, and we look for matches between candidate genes and phenotypic features. And that's the basic, quite straightforward principles of matchmaking. Now, the problem was we had people with data in systems all around the world that were not allowed to put all of their data in the same database in the same country. And this is a model that's now being used quite commonly. So the traditional model is a centralized database like dbGaP or ClinVar, where everybody submits to one database that's hosted in one place, and then everybody just puts their data there and accesses it. The future of particularly international data sharing is going to be through federated approaches, where everybody's data sits in their own institution, hosting their own database. That deals with a lot of countries have international laws that don't let patient data leave their country. But we can query those databases through APIs. So an example of that is the Matchmaker Exchange. And we now have a system that connects patients around the world. We have 7 nodes that are hosted in 4 different countries: Australia, Canada, the US, the UK, and actually just as of a couple weeks ago, Japan is now hooked up, but haven't updated the figure yet. However, there's data in there from 69 countries, so really connecting all of these databases worldwide. Thanks to Harindra here at the Broad, who developed the Matchbox system for our Center for Mendelian Genomics, we are connected to this system as well. We have a website, matchmakerexchange.org, that helps guide you in using this system. And there is now data from over 100,000 patients across these 7 nodes, from over 4,000 contributors from 69 countries. Although there's hundreds of gene discoveries that have been made throughout the individual nodes within them. If you look at matchmaking across the 7 nodes, we've now been tracking 21 papers that are in preparation from discoveries that happened once we launched the Matchmaker Exchange and could do these queries across nodes. So that's kind of where the system is. It's actually working quite well at this stage. And this is just data from our Broad Center for Mendelian Genomics. We focus on a number of different diseases as well as orphan conditions. Matchbox is our system, and as of the progress report, I think yesterday it was, we've looked at 2,620 cases, solved a quarter of them, and have novel gene disease discoveries at about 235, broken down by really good candidates, which are Tier 1. Tier 2 are phenotype expansions. That means a gene that's already been implicated in one disease, we discover that other mutations cause another disease. Or a lot of them are in it's a candidate, but there's not enough evidence for causality. So that's sort of where Broad Rare Disease Discovery and our CMG is at right now. So we're going to go back now to the framework and go through some specific examples of actually curating the evidence and scoring the points. So you get a better sense of how we go about doing this and how we have curators all around the world that are doing this in a distributed. It's not just like I sit there and do it all. So we had to come up. We have a tutorial, resources online, SOPs that guide people how to do this. So the first step is basic literature search and using resources like Omen, Orphanet, Gene Reviews, PubMed, and Google Scholar to try to get all your data. And then the first step is the genetic evidence scoring. So here we are looking at and giving different points depending on the variant level evidence. So was the variant de novo? A lot of these have little superscripts which point to the SOP and say, well, keep this in mind if you have you confirmed maternity and paternity in that case, because if you haven't, then you should score less. All those level of details. Proband with a predicted or proven null variant, so your loss of function, your frame shift, nonsense, canonical splice site variants. And we sort of guide with a default scoring, but then give a range. There's a missense variant. It's predicted by all the programs to be, impactful, or only half of them. So I'm going to give a little less points for that variant because it's just, computational predictions versus there was a functional assay that showed that variant disrupted function. You get more points for that. And that's how, sometimes we give 0.1 points if it's just computational, or maybe 1.5 if a functional assay showed that missense variant disrupted function. And that's for autosomal dominant or X-linked disorders. For autosomal recessive, evidence of there's a second variant in trans, at least one of them is de novo or a null variant, or there's two variants that are not de novo or loss of function, and different points scored for that. We also have scoring for segregation data. And so if you have a LOD score that's been published that's between 2 and 3, you get half a point if they only did candidate gene sequencing, whereas they'll get a full point if they sequence the entire exome or genome and looked at everything. The higher the LOD score, the more points using these two different methods. But it's important to keep in mind that even if you have a LOD score of 5, or usually 3 is enough to get your paper published, but that is still implicating a locus with a high statistical confidence. It is not implicating that gene, right? And a linkage interval with a LOD score of 3, typically about 100 genes in that region that all have the same level of statistical evidence that they're important, right? Because the linkage interval spans, hundreds of kb. So just because you have a incredibly high statistically significant LOD score does not mean the variant in the gene that you're looking at is actually implicated. And you need gene and variant level evidence also. So you only get, max of 3 points because that is not specifically implicating the gene in most cases. And then we get down to case control studies. So, for the rare Mendelians, you're usually dealing with individual cases with individual variants. As we get into phenotypes that are slightly more common, like breast cancer and, cardiac diseases and things like that, you then really start to need to go over to case control studies. And those could be single variant analysis, looking for the variant at a frequency in cases and that same specific variant in controls, or you're doing aggregate variant analysis, often called burden analysis, where you look at the burden of loss of function in the gene versus the burden of loss of function in controls and look for statistical correlation. And then, of course, you have to look at a number of factors of the quality of the study, race matching, the statistics, et cetera, and that leads you to score anywhere from 0, if it wasn't a good study, to up to 6 to implicate that gene using case control studies. I think I kind of already went over this variant evidence and segregation evidence. I guess I could have done the larger screen version. Okay, so I did it all in one slide. Forgot there were details in there. But here's some examples of some, case control studies. So, and they're in our SOP, so they're genericized in this case. Breast cancer, a variant found in 100 cases out of 12,000, but also observed in 7 out of 4,500. In this case, they were matched by age, ethnicity, and geographical location. The specific variant being looked at was a frame shift variant. The odds ratio that was calculated was 5.4 for risk for cancer with a p-value very statistically significant. So that was a single variant analysis, and it scored very high, point value based on the p-value and the well-matched, case control data. In this case, another study for cardiomyopathy matched by location but not age or ethnicity. The cases and controls were genotyped for the variant. It was a single variant analysis. The p-value wasn't as high, but it was statistically significant. They scored that 4 points. This was a burden analysis looking at ovarian cancer where they sequenced the gene and counted all cases with null variants and then compared that to exact population database. A p-value of 0.015, so statistically significant but not that high. They scored 2 points for that. So this is just an example of trying your best to kind of look at different types of studies and the variables and try to score points. There is some subjectivity to this, and that's again why we kind of bring experts in at the end to look at how the curators scored those particular studies and decide if they agree with the value of that scoring. Yes, go ahead.
Go back
yep.
To the slide. So you have statistical significance, but not effect size.
Yeah.
And I'm wondering, you could have a tiny effect size. I mean, if you're in a large study, of course,
yeah.
you have a tiny effect size and a small, tiny effect size and a very tiny p-value,
yeah.
which would be statistically significant, but
yeah.
irrelevant. So
and
how does that?
So I will say that we have guided that this whole approach, is only for Mendelian diseases and is not for complex traits where effect size is often very low. In that case, we suggest going over to the traditional statistical models for GWAS and not touching this whole framework.
But you are doing case control.
Yes.
Here.
So the challenge is that the world is not conveniently divided into complex traits and fully penetrant Mendelian disorders. We have stuff in the middle, right? And that's where we get, like, cancer is a good example. There are very good families with clearly hereditary breast and ovarian cancer and many other cancers. But some of the variants have lower penetrance. There are BRCA1 and 2 variants. The gene is absolutely implicated, but the variant CHEK2 is a good example. It's a cancer gene, but has much lower penetrance than BRCA1 and 2. So as the teams are looking at some of these genes where there are families, you could consider it a Mendelian phenotype, but with slightly lower penetrance. And we debate, like, should we be using this framework for this gene that most of the variants are lower penetrance, lower effect size? And, at some level, if you have any concern, you sort of throw out the scoring method and you move over to traditional statistical GWAS models looking at. And in those cases, you pay a lot of attention to effect size because that is the clinically relevant question.
Sure.
What is the effect size, right? And so this is really meant to look at Mendelian phenotypes where the penetrance, and this is where we have fights, like, well, how high should the penetrance be considered Mendelian? Like, I'll often say, well, at least 5%, but that's even pretty low. So we're not dealing with odds ratios of, 2 or less for sure. And, here's an odds ratio of 5 in this case, but so we are definitely not dealing with anything with odds ratio below 2. And in a lot of cases, it needs to be significantly higher than that. So this is, again, a semi-quantitative framework to do our best, mainly in Mendelian disease.
Heidi, just in follow-up to that, there's now some evidence, I'm thinking of Chakrabarty has evidence of the red oncogene, where you have a clearly Mendelian thing with low penetrance, but you have a common variant that is present. Now, at the moment, this wonderful system you're outlining doesn't quite take that into account. So when you cross over into oligogenicity, this issue of penetrance becomes a hugely important one.
That's right.
How is that being handled? How is it being thought about? Because it's kind of the merger of common traits and Mendelian, and yet I think more and more genes are going to wind up in that area.
Absolutely. So, an example, and BRCA1 and 2 are good examples where no one disputes that loss of function mutations in BRCA1 and 2 are causative of breast and ovarian cancer, and they lead to Mendelian segregating disease. However, there are individual variants in BRCA1 and 2 that have a much lower effect size, right? Or penetrance is usually what we talk about when we talk about Mendelian disease. Those specific variants, this is a gene level implication framework where we're looking at multiple variants in the gene, and we usually just focus on the high penetrant ones. And we ignore these, modifiers or low effect size variants and just skip them as we're implicating the gene. And then in the next question of, okay, the gene BRCA1 is a cause for breast and ovarian cancer. The gene is clearly implicated. But now somebody comes up with a variant that was thought to be benign because you see it all over EXAC or NOMAD, but you do a good case control study and you show a clear statistical increase in cases compared to controls. So that is all now in the realm of statistical assessment and looking at good studies, replicated, large, matched, all of the things that everybody thinks about in good statistical analyses. And so that individual variant is going to be annotated based on those individual statistics and is kind of considered separate from the totality of data that implicated the gene in a Mendelian phenotype. Does that make sense?
Yes. It just seems to me that your Genematcher, which is a wonderful bit of work, needs to drag with it the normal population data such that the power of that increases.
Yes.
So you got half of it. But it seems like now you got the network, you got the nodes, you have the way of taking care of the confidentiality. Why not bring in the 500,000?
It's in there. So I should say that Harinder just sent us a Slack message a week ago. You were, I think, on that too. That they have and so in the Broad Matchbox system, each of the nodes controls matching itself, which creates some challenges because they're not equivalent. But in the Broad Matchbox system, they have put in constraint scores and exact NOMAD data, these NOMAD data, to contribute to the actual scoring of the match. So if somebody throws in a gene and a loss of function variant, and in exact there is a constraint score that suggests that loss of function are not constrained, then that score for that match based on somebody sending us a loss of function will be much lower than if this constraint score is very high, suggesting loss of function is, deleterious to that gene, right? So we are now incorporating the NOMAD data and the exact data into our matching in terms of scoring those candidate genes that come in from other databases and our own variants that we've found too.
I got that, but now you're limited by a NOMAD. So you're at 130,000, but each of those nodes is 500,000 going to be in England. It would be lovely to drag those in because then your power would go way up.
So it's also a great question. Each of the matchmakers functions a little differently. So Genematcher, which is the most widely used system in the Matchmaker Exchange, is a system where nobody puts raw data. They just, you could show up, go type in your name, get up a user account. You can do this right now, sitting there, and type in your favorite gene and say epilepsy, which I did this the other day, and hit submit. That's it. Gene name, epilepsy, nothing else. So that entry right there has no data behind it that can be used in the broader set. Now, other databases like Decipher, Matt Hurls and Helen Firth, their database in the UK, which has been around a long time, has a whole set of data in there. Mostly, however, disease data, not normal populations. For the most part, I actually don't know that for sure. I'm sure they have some, but not the level that Daniel has with NOMAD, et cetera. But it is an absolutely good point, and we have talked about, in fact, the next stage of Matchmaker Exchange that we are continually discussing is one-sided matching. Like, you submit your candidate gene, and no one else has labeled that as a candidate. But maybe there's a loss of function sitting in somebody's case out there with a phenotype that matches yours. They just didn't look at it and nominate it as a gene, but it's in a database. And so what we want to do is be able to have one-sided matching where you put it in one side and it actually queries the raw data from the cases sitting in another node. The challenge there is dealing with data sharing and access and all sorts of, authentication and things like that we're struggling through, and how to do those kinds of queries, which are much more difficult than gene candidate and a phenotype. So that's where we're headed. Not only the case level data, but as you said, the normative data as well, which will help look at, bringing better race matched and things like that. So absolutely. Okay, so we're at the next stage of experimental evidence. So we actually, a group of us, Daniel leading at the time, put together a framework for gene disease causality that was much more descriptive and defined a lot of different functional categories. So we actually stole all of that from this NHGRI sponsored workshop that we had a few years ago. Actually, now it's like 6 years ago. But these are the categories: biochemical functions. So the function of A and B are the same, and they're involved, in the same disease, or the function, that example of the inborn error of metabolism I gave, is consistent with the patient phenotype, or, A is your candidate, but it is shown to interact with B, which is involved in the same disease, or the expression of the the gene is in the right tissue, those types of evidence, or functional alterations. So the gene or gene product you can show is demonstrably altered in patients carrying candidate mutations. You've got an animal model that is consistent with a human disease, and you've disrupted the gene or put in a gain of function mutation, or you've rescued. You've got a animal model that you put the functional gene back in, and it rescues the phenotype. So those are all examples of functional data that we then score in the process. And so we have these different categories of these individual types of functional data, a default point value that we've assigned, and a range, so the quality and level of how much does the phenotype actually match disease or not, can be scored. Now, we only allow all of the functional data together to achieve a max of 6 points, which only in the framework, if you recall, gets you to limited. And that's, important point, because no matter how much functional data, if you have no human case level evidence, you cannot implicate it in disease. So we sort of limit that scoring to 6, and you have to have human data to get into the moderate and above categories. So then we aggregate all that scoring, we assess the evidence, complete the summary matrix, assign a clinical validity classification, and then we have an expert who's a disease expert and ideally a researcher working in that field, as well as clinician who understands the human phenotype, work to review that and decide if they agree with what the curator put together. So that's done using this overall framework. We score the genetic evidence, the experimental. Okay, that's what I just said. I should mention, it's very difficult to deal with contradictory evidence. So we don't actually score it. We just put it in and say, yes, there was, and this is the data that's conflicting. Because some data, it might just exclude a variant and say you can't score that. Other data, like I made a mouse model and it's normal. Well, does that remove that it's a human disease gene? Maybe, maybe not. Kind of depends. In many cases, it may not. Humans and mice are different, and not every human disease gene has the matching phenotype in mouse. So it's suspicious if it doesn't recapitulate, but it doesn't totally rule it out, right? So you need an expert to really weigh in on that conflicting evidence and say, does this mean I just don't score that piece, or does it really overall argue against the data I've put together? And does it dispute it, or does it really fully refute it? If the only claim for a gene was, one case with one missense, and then 5 years later, EXAC comes out and shows that variant is in 10% of Africans, you'd refute the whole thing, right? But other examples, you may just refute one variant, and the rest of the story is fine. So that's sort of how we go about. We don't score it. We let the experts review that conflicting evidence, and they may decide it's okay, just going to ignore that piece, go ahead and score, or no, we're not going to score this. We're going to describe, give the category, and say why we think it's conflicting, disputed, refuted. Yeah.
Heidi, just before you go on to the next one, there's a couple of other areas out there of data that is brought, kind of like functional data, which you put the high-level functional data. But you have proteomic databases, you have network pathway analysis data, and you have X-ray crystallography data that is increasingly robust in terms of providing evidence that could be used in a support. How are you guys thinking about that? Because you haven't mentioned that in any way in scoring.
Yeah, God, half the time I go into these databases, I'm like overwhelmed with networks, and it's sometimes really hard to kind of use that data, and sometimes we'll engage somebody who actually made the database and explain what their network sort of thing is. Is it really implicate the gene in the same pathway? What's the evidence?
Right.
And it's
very involved.
Yeah, so we do use these databases and try to find some of that evidence, but it varies whether we feel that it's useful evidence. It's, because a lot of those data are automatically aggregated without human, like, yes, I believe this point to this point is real. So that makes it just an enormous amount of information that's hard to sift through what is good and what is not. At least that's been my experience. But, over time, those data sets will get better and better and hopefully be a little more usable. And someone can develop a better framework for how to score it. So I'm going to go through a couple of examples. We have a little less than 10 minutes left. So here's the gene PNPT1 that's been implicated in autosomal recessive hearing loss. So here's a publication with a missense variant, glutamine to glycine. The case tested negative for only the most common gene involved in disease. The variant was absent from nomad and exact. It was found as a homozygous variant in a consanguineous pedigree. So that variant got scored a half a point for just this one missense homozygous variant. Here is the pedigree where they had 3 affecteds who inherited the mutation, 3 unaffecteds who didn't, or both mutations, I should say, or the homozygous. So the LOD score in this family was 2.7, so they got 1 point for that LOD score. The experimental evidence, in this case, the variant was local, or wild type N variant was localized to mouse and zebrafish inner ear tissue. So it's expressed in the inner ear. That's good. That matches where the phenotype exists. That got a half a point. Just because it's expressed in the tissue doesn't give you a lot of evidence. There was a gel electrophoresis assay that showed the mutant protein was less able to form homotrimers than wild type protein. Well, maybe that's functionally impacting the variant, but does ability to form homotrimers, is that a biologically relevant impact? Hard to say. Only gave it 0.5 points. Evaluated the function of the variant in mammalian yeast and bacterial cells, showed it exhibited diminished function as an RNase-P RNA importer in all cell types and caused reduced viability in bacterial cells. Again, showing some functional impact, but does that mean it's going to cause hearing loss in a patient? A little hard to tell, so they scored it 0.5, so not a whole lot of data. So in the end, you had 1.5 variants for that one homozygous missense variant in a consanguineous pedigree, not a lot of data. 1.5 for that functional data, none of it was all that convincing. Got you 3 points, you're squarely unlimited. So it's at the level, if it's of a candidate gene, insufficient to say that this is a hearing loss gene. Roughly in my head, how I use these categories is things that get to moderate, I say there's a really good chance that more data is going to bring that gene up to true gene. Things that are limited, highly likely it will never go any further. That's just anecdotally how I think about these 2 categories. This was reviewed by our hearing loss clinical domain working group, which has 49 people all over the world with expertise in a variety of different types. They all agreed. SOS2 and Noonan syndrome. So in this case, there have been 5 missense variants reported in association with Noonan syndrome. The gene is constrained for loss of function variants. However, Noonan syndrome is a largely gain-of-function disease only due to missense variants in most of the genes. So the mechanism is probably not loss of function. So this data in the constraint score and exact for loss of function may not be that relevant. It is a little constrained for missense as well, but not to say devoid of missense. So all we've got is 5 missense variants at this stage. And these are the 5 different variants, or I should say 5 different probands. One of them was de novo. So here's the family with only 1 segregation, so that was not enough to score it. 0.5 variant or points for this variant. The de novo occurrence where maternity and paternity was confirmed, that got 2 points. Here's another family, half a point for that. There was then a burden analysis done with 50 Brazilian probands that were negative for the other Noonan syndrome genes. And in this case, of the 107 Brazilian individuals who didn't have Noonan syndrome, they found, 5 variants in SOS2 were identified, but 3 in the control population. So This did not have statistical significance, so this study scored 0 points. And then experimental evidence, we're running out of time, so I'm just going to really rapidly go through this, looking at ERK and MEK phosphorylation on a Western blot, that scored a 0.5 point. Looking at RAS kinase activity, that scored a 0.5 point. A null mouse was generated targeting deletion of the gene, and there was no phenotype. However, that's a loss of function model, and we think that this is likely a gain of function. So it scored 0 points. That model is really irrelevant to this question, right? So in the end, there's 7 points for those 4 or so probands, one of them de novo. Very little functional evidence, gets you 8. It's at moderate. So this is probably going to be a disease gene as more and more families, but we're not quite at that level, and this was agreed by the experts. So I think we are done. I just wanted to acknowledge a large group of people in ClinGen that are the gene curation group of which developed this framework. Also, we have a gene curation journal club led by Marina, who also helped me with these slides and all the examples, so thanks to her. So if anyone's interested in ongoing examples of gene curations, we have a group that meets, and it's has WebEx, so you can call into it from wherever, and the people take turns from all around the world in doing gene curations, and we're walking through those examples and how they're scored, etc. So if you are interested in more in-depth gene curation, please contact either Marina or myself, and we'll get you hooked up into our monthly gene curation journal club. And I will stop there. Thank you very much. And if you want part B on variant curation, come next week at 10 to 11. Thank you.